Thank you so much, and it is a great honor and a pleasure to be here today, to hear Jeff's wonderful talk, and to also share my enthusiasm for a growing awareness of the interconnections of different scientific disciplines. In this sense, it is a special privilege to be the Linnaeus lecturer, because Carl von Linné, although he's best known for his work on plants and botany, on the binomial nomenclature, saw in nature a uniformity, a system in which all parts of nature were linked. So his classification system was beyond just plants. Of course, it included animals and the kingdom of minerals. Now, mineralogists today no longer accept the Linnaean ideas of mineralogy, although we do still talk about mineral species. I think Linnaeus, in this sense, was a great philosopher, and that's the spirit which I'd like to follow him today, to try to show you some linkages between the world of minerals, the geosphere, and the world of life, the biosphere. I have to begin by acknowledging dozens of collaborators in 6 different areas of science. These individuals come from 20 different institutions around the world, and I'm going to be touching on aspects of each of these, but I just want to credit. I work on complex systems with Niles Eldredge and Jack Szostak, who you may remember a couple of years ago won the Nobel Prize. We look at a new way of thinking about minerals called mineral evolution, have a large number of collaborators in that field, and I'll touch on that today, but tomorrow that'll be the main focus of my lecture. We consider prebiotic chemistry and the origin of life. I'll touch on that a little bit later today. We also are interested in mineral molecule interactions in general, and in particular chiral interactions, the interaction of chiral or handed molecules on mineral surfaces. And finally, interest in deep life, the deep biosphere, which will end my talk today. So we're going to take a journey today, and it's all linked by the concept of evolution. I think Jeff's talk was a wonderful lead-in because he really was implying that planets undergo an evolutionary process from the protoplanetary disk to their early stages to different stages. But many different systems change over time, and that's the most fundamental definition of evolution. However, when most people talk about evolution, they mean something more. They mean that there's an increase in patterning or complex behavior or diversification through time. And in addition, evolving systems depend on what came before. So you're talking about a sequence of steps from a first step to the next stage to the next stage and so forth. And these are characteristics of all kinds of evolving systems. Now, in biology, of course, there are a couple of special additional aspects. We talk about common descent, and in detail, the Darwinian idea of evolution by natural selection by means of a genetic mechanism we now know. And these are special aspects of biological systems. What I want to do is to step back from that and look at the more general principles that apply to all complex evolving systems. And they occur throughout nature, throughout the cosmic evolution, since the Big Bang. They occur in elements in isotopes, in minerals, in organic molecules, and of course in life. And the idea here is that natural systems are intrinsically complex. Planets are intrinsically complex. And evolution occurs and is driven by selection in that complex environment, including gradients, cycles, fluxes, interfaces, chemical complexity that you find in planetary systems that you don't generally find in a laboratory experiment. There are 3 key ideas I want to get across. First, the complex systems display diversification in all these aspects, in type and behavior and patterning, diversifying through time through selective mechanisms. In all these systems, you see them throughout the cosmos, throughout the history of the universe, in elements, minerals, organic molecules, and life. And in all of these systems, you see qualitatively similar patterns and behaviors. You see radiation, you see punctuation, you see extinction. These are terms that, while very appropriately applied to biological living systems, also apply to a variety of other domains in the cosmos. And seeing the general principles, looking for empirical similarities in these wide variety of evolving systems, can then help us, can guide us in the experimental study, particularly for the origin of life and its evolution. We don't know in detail how life originated, but if we understand general principles of all evolving complex systems, that'll point us in the right direction. So, let's go through some examples, starting right with the Big Bang and nucleosynthesis. Here we see after the Big Bang, there were only a few elements, a few isotopes. I'm going to show you 4 diagrams. On the vertical scale is abundance. On the horizontal scale is atomic number. And here in this particular diagram, what you see is that following the Big Bang, there was a lot of hydrogen and some helium and not much else. But hydrogen and helium inevitably, inexorably formed the first generation of massive stars. And these stars went through cycles of fusion that produced other elements. And so you have those first, oldest stars that have an increased abundance of elements, all from fusion processes. And some of those large stars explode. And when they explode, they go through the supernova process, which gives you a rapid neutron capture process that gives you more elements. And as you go through cycles of stars, you see the contemporary abundance of elements. And we recognize roughly 2,000 different isotopes now in the 100 or so plus elements that we recognize in the periodic table. This is a process of complexification over time. There are punctuation events, such as in the explosion of stars. There's extinction as various isotopes, radioactive short-lived isotopes, disappear. You see all the characteristics of an evolving system here just in this very simple example. We're exploring a second example, mineral evolution, which will be the main point of my lecture tomorrow. So I just want to go over the general outlines and principles of this exciting new way of thinking about mineralogy. The fact is that through time, the mineralogy of Earth's near-surface environment has changed. It's changed in the terms of the diversity. There's just simply the number of different mineral species, but in many other aspects as well. The relative abundance of those minerals has changed. The compositional ranges of those minerals have expanded through time. And the grain sizes, the sizes and shapes of the minerals themselves. When you go to a mineral museum, you see the beautiful crystals of different forms. It turns out that early in Earth history, the forms of those crystals are different than they would be today. And I'll give you a punchline. It's primarily because of life. So, again, with mineral evolution, I want you to be sure to understand that I'm not talking about Darwinian evolution here. We're talking about change over time, something that's been talked about in earth science circles for centuries. Evolution of the Igneous Rocks is a famous book that does that. There's the implication here of complexification or diversification over time. And also this congruency, the idea of one stage is necessary before you can go to the next stage and the next and the next. But in spite of the fact I love this cartoon that came out shortly after our publication, we're not talking about Darwinian evolution. So crystals do not evolve. Quartz is quartz is quartz throughout geological history. But having said that, there are some important ways of thinking about changing mineralogy over time. I frequently visit mineral departments and ask them, what was the first mineral in the cosmos? And I'm greeted with blank stares. Can you imagine this? We are mineralogists. We spend our lives thinking about minerals. We spend our lives thinking about the geological environment, and yet no one had thought of what was the first mineral in the cosmos after the Big Bang. Where was the first mineral? How did it form? Well, it turns out when we began to think about this process, we realized that there are a dozen or so minerals, the precursors of all planets and moons. And they form in the envelopes of exploding stars where you have a high enough density of atoms that you still can get atom-atom interactions, but you get a cool enough environment that very high temperature crystalline phases can precipitate. And we suggest that diamond is the first mineral in the cosmos. And sharing the stage with diamond are a variety of carbides, nitrides, oxides, maybe a silicate or two, the minerals, all formed in the envelopes of exploding stars, all present in pre-solar grains, the grains which actually was the dust of the dust and gas that made our solar system. They still exist today. You can find them high in the atmosphere. You can find them in some very primitive meteorites and extract them, recognize them, and see these nanophase materials. That's the starting point. So the question of mineral evolution is, how do you go from the 12 minerals to the 4,500 species we know of on Earth today, and about 100 new ones are found every year now? So there are three processes, and these are processes that are deterministic. They will occur potentially on any Earth-like planet. The first one, separation and concentration of chemical elements. And this occurs primarily through fluid rock interactions to select and concentrate the elements, so form new minerals that way. Second one is increased ranges of temperature and pressure, the intensive conditions that will allow you to have different access to phase space, which gives you different assemblages of minerals. And finally, third, and it turns out most significantly, life. Because life is the primary engine, the driver of mineral diversification. Okay, we divide Earth history then into three eras of mineral formation, three eras of mineral evolution. And the first has to do with a process that Jeff's already alluded to, the formation of planets in that protoplanetary disk. And you can imagine that you have dust and gas in this disk surrounding the central star. The star is forming through gravity, bringing hydrogen and helium, growing larger and larger, and then nuclear fusion begins, and the star enters this very violent early stage called the T-Tauri phase, where it ignites, where blasts of heat energy come out, bathing the dust and gas, forming droplets, melting and processing and refining that dust. And in this process, the first stage, you form what are called primary chondritic minerals. There are about 60 of them. Most of them are micro or nano phases, but 60 minerals. So you've gone from 12 now to 60. And these chondrite meteorites that are still found today, that still fall to Earth, the least weathered, the least altered of these materials tell us of that earliest phase of planet formation. And we know now that the chondrites started clumping together, as Jeff said, fist-sized, room-sized, city-sized, 100, 200, 300 kilometers. And when this happens, new changes occur. You find that the bodies, when they reach about 100 kilometers, they start to melt. They have a lot of radioactive elements. They have a lot of gravitational heat. The whole body melts. You differentiate an iron core and a silicate mantle. And that differentiation process, this is the winnowing out of materials, selecting and concentrating elements, forming new minerals. And so you eventually get up to about 250 different mineral species. Some of those are formed by aqueous alteration, water interacting with the rocks inside these planetesimals. Some of them are formed by pressure and remelting and temperature. And some of them are formed by impacts and collisions, because when two of these planetesimals come together and they collide, there are tremendous shock temperatures and pressures, and a whole new suite of minerals form. And all 250 of these minerals are still falling to Earth today in the form of the meteorites that build up our collections in the great museums. So this is the first era, the era of planetary accretion. You've now formed planet-sized bodies. And what happens now is the reworking of the crusts and the mantles of these bodies. The first stage is just basic igneous petrology. When you have a melt that's silicate-rich and it cools, you have a sequence of minerals that form. It's known as Bowen's reaction series. He's the one who wrote that famous book, Evolution of the Igneous Rocks. You see a continuous series of what are called the feldspar minerals, compositions changing from more calcitic to more albitic, or from calcium to sodium-rich feldspars. You also see a discontinuous series of minerals containing iron and magnesium. And so this is, again, this is a driver of mineral diversification. And what we think now is that if you have a relatively volatile, poor world like Mercury, like the moon, these igneous processes can get you up to about 350 different mineral species. And that's probably it. It's hard to imagine any other way minerals could form on these small, essentially dead worlds. However, if you have volatiles, if you have water, then you can do other things. You can make clay minerals, you can make ices, you can have evaporites. And this gets you up to about 500 minerals. We think this is where Mars stopped. Mars certainly has more diversity than the moon, or mercury. But it's hard to see how you can get much beyond 500 different mineral species. If you're a slightly larger body like Venus, like Earth, you have enough internal heat to remelt the basalt, the rock that forms the crustal part of all of these planets. And when you remelt basalt, you create a new rock called granite. And granite is a wonderful concentrator of rare and exotic elements. And as a consequence, you have about 1,000 mineral species now, many of them in what are called pegmatites. There's some very famous pegmatite localities in Sweden. I wish someday to visit some of them. But you know that there are boron minerals like tourmaline, lithium minerals like spodumene, beryl has beryllium, and others, tantalite, polyiosite. Polyiosite's an amazing, a silicate rich in cesium. Imagine a crystal that you can hold in your hand that's a pure cesium silicate. Cesium, one of the rarest elements, a part per trillion in Earth's crust. It takes tens of thousands, perhaps hundreds of thousands of cubic kilometers of Earth's crust that have to be processed by fluids circulating through them, selecting and concentrating that cesium, and then concentrating it down to a single crystal. So the point is, there are these rare minerals, but these rare minerals take lots and lots of cycles of fluid-rock interaction, and it may take 1 billion years or more to make them. Now on Earth, we had the time, we had the energy, we had the fluids. But I doubt if Venus ever got that far. Venus is a very dry world now, so it probably didn't. And then Earth has another trick up its sleeve, and that's plate tectonics. Because with plate tectonics, you take slabs of crustal material and you penetrate the mantle. You take these wet, compositionally complex materials down, where they melt, where they reprocess. Those fluids come back up through the upper mantle, through the crust. As they go, they select and concentrate ever more elements. And so you get all sorts of new modes of volcanism, which produce new minerals. You have massive sulfide and other base metal deposits, sulfosalts by the 100 new mineral species that you would not see otherwise. You also have things like high-pressure metamorphic rocks. Because of plate tectonics, slabs of the crust are taken down to as much as 100 kilometers depth, then pop back up, and with them come all sorts of high-pressure minerals that you wouldn't otherwise see. These are the processes of mineral diversification that are possible because of plate tectonics. So, 1,500 mineral species the Earth can form in these physical and chemical processes. That's what a volatile rich planet anywhere in the cosmos, I think, can do. 1,500 species. But how do you get any farther? What other minerals could possibly form? And we come up to a roadblock, except for one thing. We now have the era of biomediated mineralogy, and life is what progresses, which allows more mineral evolution. In fact, I'll give a lot more about this tomorrow, but the short story is that life learned how to create oxygen by photosynthesis. An oxygen-rich atmosphere, and therefore an oxygenated near-surface environment, led to a radical transition in Earth's geochemistry and its mineralogy. And I would say, just in reference to the previous talk, that Earth-like planets, are not Earth-like planets until life has had 2 or 3 billion years to do its job. You would not recognize Earth 3 billion years ago. You would hardly recognize it 2 billion years old. There might have been a little bit of green scum, but the land itself would be totally barren. It would be baked by ultraviolet radiation because there was no ozone layer before life produced that oxygen. It was a totally harsh, hostile environment. You would have died an agonizing death in a matter of seconds on such a planet. But life comes along, and because of oxygen, you have thousands of new minerals that would not have formed. I'll go into more details tomorrow, but the kinds of things you see are massive banded iron formations, huge manganese deposits. These are the great ore deposits of the world. You also see minerals that would not otherwise form. Some of our favorites, turquoise, could not form without that oxygen-rich near-surface environment. Azurite and malachite, chrysocolla, other minerals, thousands of minerals of copper, of uranium, of manganese, of iron, of nickel, on and on the list goes. These minerals could not form without life first changing that near-surface environment. When did these minerals first appear? Probably within a few 100 million years after the Great Oxidation event, which itself began about 2.5 billion years ago. And then, of course, life on land. And Earth has only looked like this for the last 400 million years. Earth has only been an Earth-like planet for less than 10% of its history. It's a sort of a shocking thing to think about. So, land plants. Think about the coevolution of the geosphere and the biosphere. Land plants, because of their root systems, create clay minerals in the soils. The soils get deeper, which means land plants can grow taller. Land plants go taller so they have deeper root systems, which create more clay minerals. The clay minerals allow the plants to get taller. In a 40 million year span, starting about 400 million years ago, land plants went from being about 1 centimeter tall in less than 1 centimeter of soil to being 15 meters tall in 20 meters of soil. That's the coevolution of the geosphere and the biosphere. And of course, then came biomineralization, where animals learned how to make shells of calcite, calcium carbonate, of silica, phosphate, and other materials, and that forever transformed the surface appearance of our globe. There are profound implications in this view of mineralogy changing through time. One is that it suggests a new way to contrast and compare the different planets. Planets go through different evolutionary pathways as reflected in their surface mineralogies. So Mercury and the Moon, perhaps only 350 different species of minerals on these volatile, poor, small worlds. Mars was wet, volatiles, maybe 500 mineral species. And then you have Venus, which probably produced granites, so it may have gotten up close to 1,000. Earth had plate tectonics. It went to 1,500, and then it became alive, and we have the 4,500 species that we know of today. Mineral evolution thus points to targets for NASA. What do you look for on Mars and other worlds? Well, minerals are very robust potential biomarkers. If you see a turquoise crystal, if you see banded iron formations, you probably have a smoking gun for life. And maybe someday we'll be able to collect spectra off the surface of those distant planets. And if so, we might see distinctive mineralogy which points us to living systems. And finally, I have to say that one of the key reasons I think this is exciting is as a mineralogist, I have seen the decline of my field over the last 30 years. In the United States, as mineralogy professors retire, they're not replaced by mineralogists, they're replaced by geobiologists or environmental chemists, because mineralogy has traditionally been very much steeped in the physics and the chemistry of mineral specimens, isolated. If you go to a mineral museum, what do you see? You see a crystal formula, you see maybe the crystal group, it's hexagonal or it's trigonal, you might see a locality. But when have you ever gone to a mineral museum and seen the age of the specimen? And what could be more fundamental for a geological specimen than its context in terms of an age, a time during which it was formed? This is why mineral evolution changes the way we think about mineralogy. It adds that fourth dimension of time to our study. It makes, therefore, mineralogy part of a dynamic story of Earth history. We see the formation of planets. We see the impact of the moon and how that great impact the moon-forming event had on the history of our planet. We see the formation of oceans. We see the initiation of plate tectonics, the origin and the evolution of life. These become a central key part of mineralogy. And by the same token, as a mineralogist, I'm delighted to tell you that with this concept, mineralogy is once again returned to the very center of the Earth and planetary sciences as perhaps the most fundamental part, because after all, everything we know about the Earth comes from the minerals that we can pick up on the ground. Okay, so we've looked at nucleosynthesis and seen an evolutionary process. We've looked at minerals. We see the same kind of evolutionary process. Now let's look at prebiotic organic synthesis. Chemical evolution. What is, in effect, the geochemical process that led to life's origin? And here the same principles apply. Now, we make some basic assumptions about the origin of life. The first one is the first life forms are carbon-based. There's no exotic chemistry here. It's just simply the organic chemistry we learn, the biochemistry that we learn. The raw materials are basic: the oceans, the atmosphere, rocks, and minerals. And we see that the origin of life required a sequence of steps, emergent steps, if you will, where you're complexifying the environment step by step. Now, those steps, I envision 4 of them. The first one is making the biomolecules, the amino acids, the carbohydrates, the lipids, the building blocks of DNA and RNA. You make those. Then the second step is to assemble those into macromolecular structures. The third is that you have to have a self-replicating system of molecules. That's the big step. That's a tough one. And then the last one has to do with natural selection, molecular evolution by selection processes. And that we also understand extremely well. So let's look at these. The first step is easy. Making molecules, it turns out, is easy. Stanley Miller, Harold Urey, 1952 at the University of Chicago, used this simple apparatus. They made amino acids. They made carbohydrates. They made building blocks of DNA and RNA. That was simulating the surface of the Earth, where there's lightning penetrating an atmosphere which contains, depending on your point of view, different amounts of hydrogen or methane or ammonia, maybe nitrogen, maybe lots of different experiments. They all work. Don't let anyone ever tell you that the Miller-Urey experiment was flawed, because it turns out any combination of molecules can produce these organic molecules, some more than others, but this works. But it also works in deep space. We know that deep space is just loaded with organic molecules that fall to Earth in those carbon-rich meteorites. And our experiments look at yet another energetic, chemically rich environment, the ocean floor hydrothermal vents, the black smokers. And here, too, you can make organic molecules very easily. So a typical experiment that we would do is taking a gold tube, sealing in reactants like pyruvic acid. That's a small molecule of carboxylic acid with 3 carbon atoms. We add CO2 and water. We run this at 200 Celsius, 2,000 atmospheres for a couple of hours. And what we expected to see in this experiment, what we hoped to see, what we hoped would be the science paper that made our reputations, was CO2, 1 carbon, plus pyruvic acid, 3 carbons, gives you oxaloacetic acid with 4 carbons in the molecule. And that would be a key step in what's called the TCA cycle, the Krebs cycle. And if we could show that, then we would show that the plausibility of a Krebs cycle starting spontaneously on the ocean floor, and it doesn't work. Instead, what we got is our capsules puffed up. There was a yellow, brown, oily goo that starts pouring out of the capsules when you open them up, and then you smell that aroma, and it's so strong and pungent. You know you've made something interesting. At lower temperature, it sort of smells like molasses. At 300 Celsius, it smells exactly like Jack Daniels. And people would come by the laboratory, and they'd smell and say, is there a party going on? Can I have some? But here's the spectrum, the mass spectrum of these molecules. This is a grass chromatograph of the samples. And every one of these peaks is a different organic molecules, hundreds, perhaps thousands of different molecules. What you do is you can make molecules so easily at high temperature and high pressure in any energetic environment. The trouble is you make too much. You make way too much stuff. Okay, what about using minerals as catalysts? Here are some experiments we've done. And the key for organic synthesis in a prebiotic environment is to make carbon-carbon bonds. It turns out there are a couple of different ways of doing this. You can add CH2 groups to make a chain longer. That's what we call Fischer-Tropsch synthesis. You can also add CO groups, or hydroformylation. And what you see here is just a whole variety of different minerals on the horizontal scale, different sulfides and oxides that we used. And in red and blue are just the relative proportions. What you basically see is any kind of mineral you throw into these capsules, you do organic synthesis. The ocean floors, the hydrothermal vents, were incredibly productive of organic molecules. And then the question is, well, where does the ammonia come from? You need nitrogen in a reduced form called ammonia. And where did that come from? We can take dinitrogen gas. We can take nitrate. We can take nitrite. We put it into capsules with these same minerals. You see rapid conversion of the nitrogen-bearing compounds to ammonia. In the case of nitrate and this iron sulfide, 90% conversion to ammonia in just 24 hours. And even with magnetite, very common ocean floor mineral, you get significant 70% conversion in 24 hours. So we also find that the minerals can serve as reactants. They don't just catalyze reactions, the minerals themselves dissolve. Little parts of the minerals, iron sulfide and nickel sulfide clusters, become combined with these organic molecules in aqueous species that themselves have catalytic properties. This was an amazingly abundantly productive environment. And so the conclusion is that prebiotic synthesis of these molecules, whether it's Mill or Urey, whether it's in deep space, whether it's on the ocean floor, it was easy, it was simple, problem solved. You have amino acids, you have lipids, you have carbohydrates, whatever you want, you have them. The problem is there's too much diversity. And so, in spite of the fact that I'm delighted to say minerals played a key role here, and as a mineralogist, you ever notice that about the origin of life? People in origin of life always seem to see the process in terms of their own specialty. I'm a mineralogist, so naturally minerals played the key role in origin of life. Other people are lipid experts, and they talk about the lipid world. People talk about the RNA world. It's human nature, isn't it? But having said that, minerals played a key role in the origin of life. Step 2 is much more difficult. It's this molecular assembly. You have to select and concentrate and assemble those small molecules into larger structures. And one way we know this happens is through self-assembly. This is what happens when you put lipids in a solution, an aqueous solution. You see these little vesicles forming. So that yellow-brown oily goo I showed you before, I looked at that and I threw up my hands and said, that's just trouble. There's nothing good can come of this. Dave Deemer, a colleague at University of California, Santa Cruz, said, have you tried putting this stuff in water? So we did. We put it in water, and lo and behold, they self-assemble into vesicles. So here you see, even with an incredibly complex chemical mixture in which we don't treat it at all, you just put it in water and you form cell-like membranes with insides and outsides. Now this is certainly not the origin of life here, but this process of self-organization clearly took place. And this was part of the process. But there are many molecules that are soluble in water, amino acids, sugars. So what happens to them? And we think maybe minerals played a key role. So we have now a very intensive research project going on looking at mineral-molecule interactions. Some of our studies use rutile because rutile is a particularly stable mineral in water, and you can study it in some rigorous detail. And so rather than going into a lot of details, I'll just show you we collect data by looking at absorption curves as a function of pH. How much of a molecule, for example, glutamic acid, that's an important amino acid, or ribose, an important sugar, how much of these molecules in solution is pulled out of solution and concentrated on the surfaces of the minerals. And that's as a function of pH, as a function of how salty the solution is, and so forth. And if you do those experiments, you can actually say quantitatively how the molecule is attaching to a surface as a function of pH. Here, an interesting result, and this is very typical, is that molecules are attaching in more than one way. Some are lying down, some are standing up. And in this particular instance, as you increase the concentration of this molecule in solution, because the surface gets more crowded, more and more of the molecules start standing up. Of course, one molecule at a time doesn't tell us very much. One of the most exciting things was discovered recently, and this is unpublished data, this is just brand new, but it's an insight that I think is profoundly important. Look at different amino acids, and they absorb differently at different pHs. So lysine, for example, absorbs a little bit more at high pH than at low pH. That's because of its charge. It's positively charged, so it absorbs there. But you have very little absorption at neutral pH. On the other hand, glutamic acid or aspartic acid absorb much more strongly at low pH, but not very well at high pH. Well, lo and behold, if you put both of these amino acids together in your sample with rutile, they both absorb about an order of magnitude more. There's cooperative effects. And this is fundamentally important to understand the origin of life, because the origin of life isn't about selecting and concentrating a single molecule. It's selecting, concentrating, and then seeing self-organization above a whole group of molecules. And what we're seeing here is a cooperative effect between the lysine and the glutamic acid mediated by the mineral surface. This is exactly the kind of effect that has to take place if you're going to have complex three-dimensional molecular structures arising from a mineral template. In terms of selection of molecules, the most difficult, the most complicated thing to understand is chiral molecules. Molecules typically come, amino acids, for example, sugars, in left and right-handed forms. You've probably seen pictures of this sort where you have a left-handed and a right-handed molecule. And it turns out for biological systems that handedness is very important. Life today uses primarily left-handed amino acids, right-handed sugars. How did this selection occur? So you wonder at what point in the history of the origin of life did homochirality come into effect? And also these experiments turn out to be tremendously important because this is a huge pharmaceutical problem. Making chirally pure molecules is really important. $200 billion a year is spent to do this. Thalidomide is a classic example, where the right-handed form cures morning sickness in pregnant women, but the left-handed form causes birth defects. This is why thalidomide caused such tragic consequences some decades ago. In industry, there are two different ways of making a pure-handed molecule. One way is you start with a mixture of left and right, and you pass them through some kind of column, a column that separates chirally, and so that you get pure solutions. Another way is to start with reactants that are not chiral, but react them on a chiral mineral type surface. And so the question is, can we do this sort of experiment using natural materials? Make a long story short, some years ago I demonstrated you can select left and right-handed molecules on the chiral faces of the very common mineral calcite. Left and right-handed aspartic acid are pulled out of solution preferentially on different faces, giving you very strongly chiral molecular circumstances. Many other minerals have chiral faces. And so there's great opportunity in the natural world to pursue this. However, I want to make a point that here's a quartz crystal. Every face of this crystal is chiral because quartz is an intrinsically chiral material. It has spirals of atoms that either go to the left or to the right-handed sort of spiral. But look at this quartz crystal that I collected when I was a teenager in New Jersey. If you look closely at it, you'll see that it has a coating of iron oxide. But only certain faces are coated. This underscores the fact that each interaction of a molecule and a surface is very specific to the structure on that surface. What everyone had done prior to our experiments is to take quartz crystals and crush them to as fine a powder as you possibly could get. And that increases the surface area. But by doing that, you lose all of this specific structural information which is present on the single crystals. Consequently, what we do is we look at single crystals. We look at natural crystals that have chiral faces, like this feldspar, the commonest rock-forming mineral, diopside, the commonest mineral of magnesium and iron, calcite, again, my favorite of these minerals, because it turns out these chiral faces have very, very strongly chiral structures, including grooves along which molecules assemble. You not only select left versus right-handed molecules, but you line them up in exactly the way you need to form polymers, polypeptides. And we can do density functional theory calculations, this work with my colleague Aravind Asthagiri, where he showed that the right-handed molecule on a left-handed surface has 2 points of attachment, but the left-handed molecule has 3 points of attachment. That's the chiral selection. That's the bowling ball problem. Your right hand can't use a left-handed bowling ball, the big American-style bowling balls. And so in this particular case, there's an 8 kilocalorie per mole difference in the absorption and the binding energy of these two molecules. That's a huge difference. This is a natural chiral selector in nature. Okay. The trouble is life is short. Graduate student careers are even shorter, and there's so many minerals and so many molecules, so very many molecules in so little time, and so what do? So we've been developing a combinatorial approach where we use microarray technology to array, in this case, various left and right-handed pentose sugars on a feldspar surface. You can then put this in what's called a TOF-SIMS, a time-of-flight secondary ion mass spectrometer. I can tell you more about that if you like to hear about it, but basically you can image each of these spots. You can see what adsorbs. You can do mass resolution on them. Here at mass 43, we see that the sugars have a peak at 43.02. The amino acids have another peak at 43.04. You can resolve those, and you can tell what sticks and what doesn't. As simple as that. So this is a wonderful combinatoric approach to understanding mineral-molecule interaction. The conclusion is molecular selection can occur by two different mechanisms: self-organization on the one hand, absorption on mineral surfaces. On the other hand, we know that both must have occurred in the prebiotic world, and that's a key to understanding this step in the origin of life. And then the big problem: how do you make copies of a molecular system? How does it copy itself? Self-replication. Well, I think the key here, and the reason that we've not been successful so far, is that natural systems have incredibly complex environments. Geochemical systems have gradients. They have cycles: day, night, wet, dry, hot, cold. They have fluxes of different fluids. They have many, many different interfaces, including mineral surface, but also liquid gas interfaces. And there's huge chemical complexity. And all of these different properties will contribute to a robust and complicated system that samples many, many different combinatorial possibilities. Remember, on a planet like Earth, you have literally hundreds of billions of square kilometers of reactive mineral surfaces. And you have hundreds of millions of years to try this out. And if Jeff's correct, there may be millions of Earth-like planets to add even more combinatorial richness. If there is a solution to the self-replicating process, this will be discovered. There are three competing main ideas out there right now. One is that it's something like the Krebs cycle, the TCA cycle, that happens naturally. We've actually, in the laboratory, managed to get 9 of the 11 steps to work. But 2 of them yet seem thermodynamically blocked, and we're not sure how to get around that. But that's a possibility, or some modification. Another possibility is Stuart Kauffman's idea of an autocatalytic network, where there's not 10 or 12 or 100, but thousands of molecules that form a network that catalyzes itself, reinforces itself, and everything else becomes food stock. So that you basically winnow down the number of molecules gradually, gradually, gradually. It's a kind of self-replicating network, because it includes more copies of the things that are within the network and deletes the things that are outside the network. That's another possibility. There's also the idea of the RNA world, that the first self-replicating molecule was actually something like an RNA that made copies of itself. A wonderful solution if only you could make RNA easily in the prebiotic world, and nobody knows how to do that. So that remains an open question. But at some point, we know there had to be a self-replicating molecular system. And when that happened, clearly you're going to have mutations. All these organic molecule systems have much diversity with free energies of different variants very, very close to each other. And in such a system, you get competition and selection. And just to underscore this point, I want to go over something many of you will have seen before, but if you haven't, this is absolutely riveting science by Jack Szostak and his team, developing molecular evolution. These are called aptamer evolution. What Jack does is he starts by making a random series of 10 to the 16th different strands of RNA with 100 letters. So that there are 4 to the 100th power possible combinations, and he just randomly creates about 10 to the 16th. That's out of the 10 to the 60th or so possibilities. So it's just a small fraction of everything that's out there. But some of these, it turns out, will react by binding to a molecule that has been pre-stuck to the side of little beads in a beaker or in a test tube. And so what you do is you pour the solution into the test tube, you let it sit there, you shake it. Some molecules stick, most don't. Then you pour out what's left, and the only thing that's left in the test tube are these beads that have a few of these very specific RNA strands. You then go through a whole process, classic molecular processes of replicating those RNA molecules that have stuck. You collect them, you replicate them with errors. You produce 10 to the 16th new strands, but in this case, all of those 10 to the 16th strands are daughters with mutations of the original ones that stuck well. And when you do this again, it turns out that some of those molecules stick much better. Much better. And if you just do half a dozen cycles, every single time you zero in on the same almost ideal solution, the tightest binding you can imagine between the RNA that wraps around the target molecule. This is molecular evolution. Look what it involves. It involves chemical complexity. That's the random pool. It involves interfaces. It involves fluxes. And it involves cycles. These are the things that drive evolution. And I am sure that if we want to show the origin of life and the evolution of life in a laboratory, we're going to have to apply these exact same ideas. So my conclusion is geochemical complexities are key to modeling life's origin. Gradients, cycles, fluxes, interfaces, chemical complexity, these are all a key part of it. And I would have stopped there, except that there's been another potential discovery, a suggestion, a hint of something incredibly exciting. And this may be a way to solve the question of life's origin. There are now hints of something called the shadow biosphere. It was predicted some years ago, but the evidence is only just now coming to light. And this is part of the work I do with something called the Deep Carbon Observatory. If you're interested in aspects of this, please go to our website or see me afterwards. I can tell you more about it. But the idea is that both from deep drilling and also studies of very high temperature black smokers in the ocean floor, there are now hints that there is something going on above 150 Celsius. Some self-organization, if you will, of molecular systems. The formation of what look like biofilms. There's not cells. But in control experiments, it appears that these films are growing at temperatures of 250 Celsius and above. Now, I'm not arguing that this is a life form or that it's quasi-life or life, but it is some fascinating organic chemistry and perhaps pre-biochemistry. And if it's happening at 250 Celsius today in these environments, then it certainly was happening a long time ago. And if it is some kind of self-replicating chemical system that's spreading on mineral surfaces, using mineral surfaces as the energy source, then this may be telling us something about a chemical environment from more than 4 billion years ago. Because at 250 or 200 Celsius, this is above the temperature at which cells can be predators. And so you're basically safe from the life forms that could otherwise eat any evidence of this. So one of the things the Deep Carbon Observatory wants to do is try to recover some of these very suggestive biofilm-like systems and try to characterize them and see if they could indeed be a step in the origin of life. If this is true, there might be life living at depths of 40 kilometers within the Earth or even deeper in some subduction zones. An astonishing concept of thinking life 40 kilometers down. A deep domain? We don't know. But it's certainly something that is exciting and worthwhile checking. So for more information, here's the DCO website. And I just want to conclude with those 3 key ideas. We've seen that complex systems, they display this diversification over time in terms of their patterning, in terms of their behavior, in terms of just the number of species. And in each case, it occurs through selective mechanisms. That these systems are observed all around us all the time. We see them in the formation of isotopes in the elements. We see it in the mineral world. We see it in the organic chemical world. And of course, we see it in the world of the living. And finally, all of these systems display qualitatively similar characteristics. And these are characteristics that can guide our search for the origin of life on Earth. With that, I thank you very much, and thank you for the opportunity to speak today. Good.